Hello, histology class. This is Dr. Ogilvy, and I'm just going to give you a kind of a guidance on two virtual slide specimens that would be similar to what you might see on the midterm exam. Many people, when they approach this, I don't think are actually examining and thinking about the lab content and lecture content, integrating information, and putting your thinking caps on to try to pull information and synthesize it in interacting with a specimen that sometimes just asks you a straight identification, other times it asks you what the some process, like this question. So the first thing you need to do is to click on the question in the BioLucida window and click Go To make sure that you are seeing exactly the way we set it up when we made the question. And then the question said, what process produced the group of cells within the circle? So you have to think, well, what are these cells? Well, if you're stumped, then I think you have to think about what are the features here. Well, I see dark staining round structures, which suggest to me they're nuclei. I see, and you know all the staining, unless we tell you it's different, is H&E. So I see a slightly eosinophilic area out here, and then I see basophilia around these, what I presume to be cells. There's maybe two cells here, and another cell here, another cell, and maybe another cell here. The nucleus is not shown. So it looks like you have a cluster of cells surrounded by what appears to be a basophilic matrix surrounded by eosinophilic. So you have to think about, what you have learned. And then you have your reference image over here. And so you can zoom out and see the context. You see, there's where we're asking the question. And you look at it, and hopefully you recognize that this is a epiphyseal plate here, and there's bone formation going on. So just looking around, and of course this is the epiphysis here, and there's bone being deposited around cartilage matrix here. So now back to what we're asking you about, these cells. Well, the location of this clearly is in the epiphysis of a bone, and it has to be hyaline cartilage, and these are isogenous groups. So how do these cells what process produced these groups of cells? And the answer is simply mitosis. Okay, so that's just an example of going through a reasoning process and drawing on information you've learned. You need to not just make a snap identification or a snap response, but think, because that's why we give these unknown specimens in the part B of the quizzes as well as the midterm. So now I'm going to pause this and go to one more example. Okay, here's another question. First, I'm going to make sure that it's oriented the way we put it. It says, examine the tissue within the reticular box. This blank, we're asking the tissue, forms the blank. What layer of what organ? And so I'm not going to give you the answer to this, but in order to identify the tissue, first, you've got to zoom in and look for the features of this tissue. Once you recognize this tissue, then you're going to be able to answer the other question. But sometimes I think that you don't really look around carefully and notice the features that are presented to you. Now, there are students in this class that are doing extremely well in thinking through this process, but many of you are struggling with Part B because it's simply, I don't think, taking the time. And of course, if you haven't studied thoroughly, then that's also a problem. So I'm going to pause this and do one more question, which I will provide the answer. Okay, here's another one. It says, first of all, I click on this and click on Go To. Now I'm gonna reset this one because it's jumping up too high. But anyway, if this happens to you, you just simply zoom out like this, and you've got to examine. You're given that this is a part of a vessel wall, and so Hopefully you recognize that this is adipose tissue out here, and you've got some small blood vessels, like this is an arteriole here. This is getting very close to a capillary. Um, but you're asked about this vessel where the X is in the lumen. So you've got to examine the wall of that vessel, and when you see with H&E all of these very red stained, crinkly or wriggly lines here, those are elastic. You might think they're elastic fibers, but they're really elastic membranes, and there's only one type of vessel. It's the elastic arteries, and also called large arteries. One example is the aorta. Another example would be part of the carotid artery. And so you have multiple layers of these, and the dominant feature is the media that has all these layers of elastic membranes, whereas and the endothelium is very thin right here, and some of the endothelium is that is the intima. Some of the endothelium of the intima has been torn off. But here you can see the flat cells of the endothelium, and there's some extravasated red blood cells here. But the dominant feature are the elastic membranes in the media, so this has to be either a large artery or elastic artery, or you could call it an aorta. All right, I hope that that is helpful. Just remind you to take advantage of the Zoom feature, look around, see the context, and hopefully synthesize what you learned in lecture and lab, think about it, and come up with a correct answer.